Hello, friends, very good morning. Today we are going to overlook the genetic interaction or gene interaction among themselves. While studying the gene interaction, we are we have studied in two types: allelic gene interaction, and the second one, that is the non-allelic gene interaction. As far as we are studying now non-allelic gene interaction. In the case of allelic gene interaction, we got that incomplete dominance as well as the co-dominance. What we saw that in case of co-dominance, neither the recessive one neither the dominant one was expressed in F1, but the ratio in the F2 generation, what we got, phenotype and genotype, that was equivalent, that is 1:2:1 with regards to the color coat of the cattle. Well, in incomplete dominance, we saw and studied the a plant, that is the Mirabilis jalapa plant, and in that case also what we saw that the parental and the maternal color of the flower was not appeared in the F1 generation, and we got the red color of the flower in F1 generation. Though at the F1 generation they were allowed to self fertilize or pollinized, and in F2 generation what the progeny was observed that they got 1:2:1 ratio in the form of genotype as well as in the form of the phenotype. Well, while studying the non-allelic gene interaction, what we saw that in non-allelic gene interaction, the independent genes located on the same or on different chromosomes interact with each other for the expression of a single trait in the organisms. Well, while studying that, we saw the complementary gene action. What happens in complementary gene action? Two or more than the two genes, nature in the dominant occurring on the different loci of the chromosomes or different chromosomes, they interact with each other, producing the character, but neither of them produce the characters in the absence of other. What was the character produced by these were that both the white flower were crushed, and what we get in F1 generation, that the red one we got. And after allowing the red flower plant for self-pollination, and as we took the F2 generation, what we saw that there was the 9:7 ratio. Phenotypically, red flower were 9 in numbers and rest were the 7. That means what we saw here in complementary gene action, that what were the white flower were not dominant in F1 generation 2 as well as in F2 generation. That means they were suppressed somewhere and the genes were not allowed to express in this situation. Okay, the second term, that is the epistasis. What is the phenomenon? Epistasis, an allelic pair suppresses the activity of other gene. Look out, the pair of the alleles which suppress the activity of the other gene, they are found on same location of the homologous chromosomes. This allelic pair which suppresses the activity of the another gene which are found on the same loci of homologous chromosomes and they are to be known as the epistates. Now it is the other part, that the epistates, which the gene whose expression is inhibited, the gene whose expression is inhibited are known to be hypostatic gene, and the gene that inhibit the expression of another gene is called as epistatic gene. The gene whose expression, look out at these two terms, hypostatic as well as the epistatic. In this hypostatic case, the gene whose expression are inhibited, somewhat some breaks are there to express, and that condition is called as the hypostatic gene. In some cases, the gene that inhibit the expression of another gene, and in that case, when the gene inhibits the expression of another gene, that is to be called as the epistatic of, or condition, that is epistatic gene. They are of two types, dominant epistatis and recessive epistatis. Well, before touching these two aspects, we repeat the phenomenon of the epistatis, that an allelic pair suppresses the activity of another gene. Okay, the allelic pair which suppresses the activity of another gene, and they are found on the same location of homologous chromosomes. What? Again, the gene whose expression is inhibited, then they are to be hypostatic, whereas the gene that inhibit the expression of another gene, then they are to be epistatic gene. Now, the phenomenon epistatis has two types, that is dominant as well as the recessive. We will touch the first aspect, that is the dominant one. When the dominant allele of one locus prevent expression of one or more alleles at another locus, it is known as dominant epistatis. Look out at this. Well, we found the best example of the summer squash and the color of the fruit. But what we see here, the allele which are dominant at one location of homozygous dominant or heterozygous dominant, these dominant alleles prevent expression of one or more alleles at another locations of homozygous or heterozygous. This prevent expression, and it is known as dominant epistatis. Look out at this summer squash. You can see the color of the fruits which are seen over here. See, this is yellow one, this is green one, and this is some one greenish white one. So well, now we take down the example in the dominant epistatis. What we find here, the ratio of complementary, that gene is 9:7. The ratio of codominance is that 1:2:1. The ratio of incomplete dominance is also 1:2:1. But when complementary gene action shows the ratio, that is the 9:7. What about epistatis? In epistatis, what we saw, that the dominant allele at one location prevent expression of one or more alleles at another locus, that is dominant epistatis. Well, while putting the experiment of this dominant epistatis, take the phenotype, that is the white one is crossed over the green one. What we see, that the allelic gene, that is capital W, double W and capital double G, was crossed with the smaller double W and the smaller double G. This is the genes which are present in these plants for this color. Okay, these are the two allelic or pair of a genes. Well, while crossing this, what happens? This one W will come here, that is the gene, this one W from this side, and this G1 from this side. These two, that is one gene of capital W and one of from one pair, one G, were crossed with the smaller G1 and the smaller W1. So when we got the progeny in F1 generation, that we saw that white color was seen in the plant. Though the plant show the white color in the summer squash, but what was the genotype of that F1 generation? Look out at this. There were 4 alleles in the F1 generation plant. This one white, capital W, small w, capital G and small g. So when we put this, when we saw that these are the in F1 generation, what we see that when we allow the F1 generation to self-cross, we see the phenotype, that is white one is crossed with the white one, because both the white characters are put in the F2 generation for the fertilization. There is somewhat little difference in the complementary gene and in this epistasis, dominant epistasis. See this dominant epistasis. Now what happens? We bifurcate all this gene and put this in the Punnett square. This is what we call, this box is called as the Punnett square. This is the Punnett square. So, well, by forgetting this, what we see, these 4 characters of a female one are put from this side, the genes, and the male one are put from this side of the Punnett square. So this one is crossed over this one. So what we got, the phenotype and genotype in the plant, absolutely the white one, and the genotypes are of the pure parents. Look out at this, capital double W, capital double G. Okay. Again, when this is crossed with this one, means capital WG is crossed with a capital W and smaller g1. Now here, though we get white one, but what is the genotype C? Little difference, that is the smaller g has been appeared here. Capital double W, capital G and smaller g1. There is some little allelic gene changes in this plant, this white one plant. Though it is phenotypically same, but genotypically there we found somewhat difference. Simultaneously, this is again crossed with this one. Now, capital WG is crossed over smaller w and capital G. Now look out at this. What we see here, both the genes of white one, that is both the letter, there are small letter, that is W, both the small, though the green G is capital G. Genotype is capital G, double G, though, but the phenotype is white one. Same at this one also. Capital W with the small w, capital G with the small g, though we get the white one. And same as, look out at this. This one is white, this one is white, this one is white, this one is white. What we get here, 9, 10, 11, 12. All these are white one. When we look out at this genotype, what we see that when we got the 12 white one and 3 yellow one. So this is the yellow one. Okay, look out at this yellow plant. Well, phenotypically, it looks the yellow. But what about genotypically? When we see genotypes, the genotype of this yellow one and this white one are the same. So look out at this. Both the capital G are present. Both the smaller W are present. But now when we talk about this yellow one, the genotype, see the smaller letter of a W and only one capital G is seen over here. So same condition where we see the same condition we see in this yellow one. See, smaller double letter W with capital G with a smaller G. Though the green pure one, that is this one, the last box, what we see, smaller double W and smaller double green, that is the parental one. So this is the parental one which is seen in this green one. This was also the parental one which is seen over here in the first box. Though they are white and green, rest of them, though phenotypically characters of genes has expressed in the white, but the genotypes are most of the difference and the characters change between the yellow. 3 yellow are found in the one and one is the green. Well, here white, yellow, and this is the some corrections. This is not the white one, this is the green one. That is 12:3:1 ratio. So friends, this is what about the dominant epistatis. Today what we saw. Well, one has to understand the phenomenon. These are the summer squash. You can see the green one, yellow one and white one, how they, the same species, same progeny, how they got the genes action due to the actions, how we got the 12, one white and 3 one that are the yellow and one that is the green one. That is the ratio is 12:3:1 in the dominant epistatis. And what is the functions of the genes in this condition? The functions of the gene in this condition is that in dominant epistatis, the dominant alleles at one locations of homozygous dominant or heterozygous dominant, this prevent expression of one or more alleles to another location. That is known as the dominant epistatis. So with this we stop our today's video. Rest of the part we will going to study in our next video. Till then please revise it and